as kind of quickly as I can, I'm going to summarize a lot of what RU54 has been focused on, and how we really got to thinking about whether concentrations of estrogens are elevated or reduced, and that might be in the context of better understanding male-female differences, or that might be useful for trying to understand differences that occur specifically in females over the life course. So I'm going to start with some acknowledgments. I get to go to work with great people. I'm going to, talk about data from many people, but Hansel Park and Anna Yin have really led a lot of our human immunology work in the lab. And then I'm going to talk a good bit about some of the graduates from the Klein lab who initially started a lot of the animal work. So Mark's going to have to sit tight and bear with me as I argue that there are some things that we can do a little bit more readily still in animal models. And one of the things that we've been able to do rather readily with animal models and mirror some of this in people has to do with trying to better understand sex differences in vaccine immunity and responses. And so one of the aspects of RU54 had been really showing mechanisms for how it is that adult females mount greater antibody responses and are better protected when challenged than males following vaccination. We've done a lot of this work with the flu vaccine, and so I'm going to stick with that today, but I am going to tell you we've also done a good bit of work with COVID-19 vaccines. And a lot of our animal work was really pointing us toward estrogens as being critical. So I'm going to give you just some snapshots. What does this look like? So if we take mice, adult male and female mice, we vaccinate them with flu vaccine. In this case, is a 2009 H1N1 vaccine. We do a prime and a boost. This is all intramuscular. What we find are that females tend to mount greater antibody responses. We can do this a couple of different ways. One of the ways is to look at these neutralizing antibodies. So these are the functional antibodies, as we were talking about proteins. These are the antibodies that actually are going to block the ability of virus to infect susceptible cells. And something we can do a bit more readily in mice, but we could do in organoids as well, is we can challenge them. So we can take our vaccinated animals, where females have these greater antibody responses, females the open, males the closed bars, and we can challenge them with live virus. And we can, in a lab with reverse genetics, and I do this with my colleague Andy Pekos, we can start to drift our viruses. So we all know that the viruses that we get infected with are often not a perfect match to the viruses that are contained in the vaccines. We hear this all the time about the mismatch. We can do that in our mice. And even when we do that, we find that females, again, the open symbols, and we look at viral titers in the lungs, vaccinated females, they clear the virus much better. And much faster than vaccinated males. This is telling us that this vaccine-induced immunity is really doing a pretty good job protecting females much better than males. We have lots of ways that we can interrogate a sex difference. So if we're interested in the hormones, maybe we eliminate the hormone that we think is important. If we don't know that it's hormonal, maybe we're interested in trying to better understand, is it sex chromosomal or is it hormonal? And so we have lots of different models in my group that we use. And so let me just introduce one that I haven't seen discussed here today, the 4-core genotype mice. And in this mouse model, the SRY gene, which is encodes for testis determining factor. So this is really critical in early embryonic development at allowing this bipotential gonad to develop into a testis. And so what's happened in these transgenic mice is that the SRY gene has been deleted from the Y chromosome, and it has been transferred onto chromosome 3, so it can now be autosomally inherited. So this allows us, if you take this transgenic male and mate him with a wild-type female, you can have offspring that are one of 4 genotypes. You can have XY animals that do not inherit the SRY gene and therefore have ovaries. So now you can have an XY individual who has ovaries and secrete high concentrations of hormones like estradiol. You can have XY mice that inherit SRY and therefore have testes. You can have XX mice that do not inherit SRY and therefore have ovaries, and again, that high secreting levels of estradiol. Or you can have XX mice that have SRY and have testes and therefore have high circulating testosterone concentrations. So now we can just simply do a two-way analysis of variants and try to tease apart, well, what's due to your sex chromosome complement versus what's due to the hormonal milieu of these mice. Okay, so purple is animals that have ovaries, green animals have testes, okay? So what I want you to see is now if we take these mice and we vaccinate them, do that prime boost with the flu vaccine, and we look at those neutralizing antibodies that I told you about, those functional proteins, what we find are that if animals have ovaries, they have greater antibody responses to the vaccine than animals that have testes. And we can look at those B cells. We can go into those draining lymph nodes, and we can look at the cells, and we can look at things like germinal center B cells, which are going to become our long-term antibody secreting cells. We can look immediately at plasmablasts. These are going to be those antibody secreting cells early on. And what we find are that animals that have ovaries, again, animals in purple, they have higher frequencies of these cells. So not only are they making more antibody, they have more of these B cells. And now we can take these animals and we can challenge them with live virus, and we do that intranasally. And then we can measure virus titers in their lungs. And animals that have ovaries, regardless of whether they have 2 X chromosomes or an X and a Y, they clear the virus better. So these are just, ways that we can utilize animal models, not just alone or in isolation, but to start to help us better understand and hypothesize about mechanisms that could be driving sex differences in outcomes from vaccines. So this really got us thinking about estrogens. This really got us thinking about estrogens and what could estrogens be doing. The other thing that we do is we do a lot of aging studies, and I'm going to get to that. I'm skipping over that for our mice, but I'm going to come to that for a lot of our human studies. So we really started thinking, what are estrogens doing? And I know that Dr. Davis introduced Ross and really starting to think about mitochondria. We too were thinking about mitochondria. I had a really talented postdoc in my lab for quite a while, Laura St. Clair, and she came to me with a metabolomics background. And we started to think about how estrogens might be changing the metabolic activity inside of B cells, okay? Those antibody secreting cells. And we have lots of ways that we've done this, but one of the ways is to use flow cytometry to do intracellular staining to look for these metabolic factors. And so we can look at metabolic factors associated with fatty acid oxidation, mTOR signaling, mitochondrial activity, even glucose transport. And we can do this in males and females. We can do this a lot of ways. And here what I'm showing you are data for males and females who are either gonadectomized, we take out their ovaries and we can either then treat them with estradiol, abbreviated E2, or an empty pellet, so they're not getting E2. We can take male mice and we can take out their gonads, and we can either treat them with testosterone or an empty pellet so that they don't have testosterone. And now we can look at lots of different classes of B cells. So this kind of gets into the depths of immunology, all these transitional, marginal zone, follicular zone, germinal center, plasmablast, all these different types of B cells, okay? And what I want you to see, and here's the key here, you know, if it's going toward red, we're seeing greater amounts of these enzymes intracellularly in these B cells. And what you can see, whether it's for fatty acid oxidation, mTOR activation, what we find is that in the presence of estradiol, we're seeing greater metabolic activity inside our B cells than in the absence of estradiol, and that would be along this path here. We don't see as much of an effect with the presence or absence of testosterone, and we keep seeing that, and we've published that over and over again. We're really not seeing that testosterone, either the presence or absence of testosterone, is really driving these differences. And so you can kind of see this down here for mTOR signaling, presence of estradiol, really driving an increase in this enzyme, in this case, this PS6 associated with mTOR signaling. So we were really interested in mTOR as a master regulator of B cell metabolic activity following vaccination. So we've heard a little bit today thinking about, you know, so you see these types of differences, how can you then use these things therapeutically? And our SCORE, funded by the National Institute on Aging, we were really thinking about how can you use what you learn about estrogen signaling, not always to think about how do you replace estrogen, but how can you actually change the metabolic activity inside of cells? And so we did that, and just to kind of show you a little bit of this, if we take females, either adult or aged, turns out that these mice, they'll live about 2 years, so aged, according to Jackson Labs, about 17 to 18 months of age really corresponds with being 65 years or over in terms of longevity and fecundity. Mice don't go through menopause, but their ovaries really do start to go through a cessation of activity, and you do see a drop in estradiol levels as an example. And so if we take these adult and aged females, or we take aged females and we give them estradiol exogenously, what we find when we look at that mTOR signaling factor, we find that with age and a loss of estrogen, you get a reduction in this mTOR protein inside of B cells. But if you give aged females estradiol exogenously, we can bring that back. And this really corresponds quite nicely to what happens to their antibody responses post-vaccination. If you give aged females estradiol, we can elevate those antibody, including those functional neutralizing antibodies, back to levels that are consistent with adult females. We can determine, okay, well, so now we know the ligand, but as we've learned and was discussed a bit earlier with thinking about these estrogen response elements and the binding to hormone to their receptors, we can manipulate the hormone receptor. So rather than manipulate the ligand, we can use agonists that are specific for certain receptors. So I give you my little cheat sheet down here. So if we take aged females and we either just treat them with vehicle, or we treat those aged females with an agonist for estrogen receptor alpha or an agonist for estrogen receptor beta, it's really that agonist for estrogen receptor alpha that causes that increase in metabolic activity inside of these B cells and improves the antibody response, whether we look at class-switched IgG or we look at those functional antibodies. So we really can start to hone in on what is it about estrogen signaling in those B cells that's contributing to that greater antibody production. If you're not convinced it matters there, well, we can go a step further. We can take these animals, not just vaccinate them after treating them either with placebo, that's P, or with estradiol, abbreviated E2, again showing you that if we treat aged females with estradiol, we bring their antibody levels to the vaccine to within levels consistent with adult females that either have endogenous or exogenous estradiol. But now we can take these animals and we can challenge them with that live virus, those drifted viruses I told you about. And our mice can't tell us how they feel, but I can tell you that mice that are infected, they feel no better than us with flu. And so aged females that do not have estradiol, it's not that the vaccine doesn't work, they are not dying, but they do get sick and they stay sick. But in aged females who've been given estradiol, they're indistinguishable from adult females. So giving aged females this estradiol actually is not just raising their antibody, not just improving the metabolic activity within those B cells, but it's actually protecting them from disease. And that is what these vaccines are intending to do. So how do we take these types of very simple mouse experiments and start to translate them into the complexities that Mark described with humans and the heterogeneity that we see that Mark described in humans? And that's where the complexity really starts to occur, and that is what our U54 had been focused on. And so we did a lot of work showing that sex differences in immunity, as well as even adverse reactions to vaccines, are greater among young adults, so if you will, reproductive aged individuals, than what we were seeing among older individuals, 75 years and older. So what does this look like exactly? Well, going back to some of that heterogeneity, an observation that we've consistently made is that among young adults, so that's what's in these darker colors, 18 to 49 years of age, again, if you will, reproductive aged individuals, an observation that we have consistently made and consistently published on is that the responses and whether we see sex differences in the immune responses to vaccine antigens depend a lot on the antigen. And where we tend to not see a sex difference is when individuals start out already having really high antibody responses. And we have really high antibody responses to one particular component of the flu vaccine, the H1N1 component. And H1N1, that's what I have pictured here, since 2009, even out to today, even in this most recent 2025-2026 season, it's still 2009 H1N1-like. So we keep getting repeat vaccinated over and over and over and over again. And so when we look at our neutralizing antibody responses, even prior to getting the vaccine, everybody is seroprotected. That's at 1 to 40 cutoff. That's what the WHO tells us. That's if you've hit that cutoff, you're gonna be fine. You're what we call seroprotected, and that's the proportion of individuals who are seroprotected. We see a little bit of a bump up, okay, after post-vaccination, 28 days later in these people, but it's not a lot. But take these young adults and measure their responses to the H3 component, and that's what's here. And I should have introduced this. This is just mapping to show you the mutations that have occurred in this H1N1. This is the HA protein. We don't see a lot compared with the H3. This has a lot of mutations that have occurred since, in this case, 2015. So from 2015 to 2019, here are all the mutations. So what we find when we look at H3N2 is that people have pretty low antibody responses prior to getting the vaccine. Only 28% of the men, 63% of the women were what the WHO would tell us was seroprotected. But you see a real bump up. And what we also see is that when you're going from a low level of pre-existing immunity, you actually see the sex difference, where now we see that females have greater antibody titers as compared with males. But this is limited to our young adults. When we look in our older aged individuals, not only do we not see the sex difference among individuals with high pre-existing immunity, but even among individuals with lower pre-existing immunity, we lose that difference. So it's really among our reproductive aged individuals, even among humans, where we see these sex differences. But these sex differences are most pronounced when we can capture a de novo response rather than a memory response. So one of the other things that we've done that actually had never been done before is to not just measure the antibody responses to a vaccine, but to actually look at this in connection with steroid hormone levels. And so we do this by mass spec. We measure lots and lots of different steroids, their precursors, their metabolites, and I have a few of those listed here. 17-OHP is a derivative of progesterone and is very important in the biosynthesis of androgens like testosterone. And what we find is that, sure enough, among young adults, we find that males have higher concentrations than females, and we see that also among older aged individuals. Here's testosterone downstream. Males have greater concentrations than females. But you can see that estrogens, including estrone, which is upstream of estradiol, it's really the young adult females that have greater concentrations than either males or our older aged individuals. And what we can do is not just ask questions now about, well, what are the predictors of that downstream, that functional neutralizing antibody that I've described? Yes, we're interested in all the immune cells that I've described in mice. We do all that in people. But we can start asking, does your hormonal milieu matter? And could your hormonal milieu at all be important in predicting that neutralizing antibody response, how well you seroconvert, those correlates of protection that the WHO tell us are so important, but that we might even debate a little bit later. And so we put that into predictive models. And here we're looking at random forest models. Here we're looking at that model, the accuracy in young adults, and these models do actually and are more accurate in our younger adults than in our older adults. And what we find is that when you look at the top hits, not surprising, flu-specific CD19 B cells, flu-specific antibody secreting cells, super important at predicting that antibody response 28 days later. But look at these other top hits, 17-OHP, estrone, and testosterone. These concentrations of hormones seem to be very important predictors of how well an individual, especially a young adult, is going to respond to the vaccine 28 days later. And nobody's ever really thought about how hormones could be used in predictive modeling in this way. So what's going on early after vaccination? We started to ask those questions, and what's going on in those B cells? Not only are we interested metabolically, but we're interested in this transcriptionally. And so what we've found when we've done single-cell RNA sequencing prior to vaccination, and this is all B cells that are being isolated from peripheral blood, but 7 days after vaccination, so that's when those plasmablasts, those early antibody secreting cells are most active, and 28 days later in our young adults and older adults, purple is female, green is male. What we really find when we look at these PCA plots is that we really start to see a division that is limited, this division between males and females, at 7 days after vaccination. That's where we find that the transcriptional activity of our B cells really starts to diverge, particularly among young adult men and women. Something that we only see among the young adults, not among the older adults. And so you can see some of the top hallmark enriched pathways that are listed here. Purple indicates significantly greater enrichment in B cells from females as compared with males. Green is going to indicate greater enrichment in males as compared with females. And so the top hit that keeps coming up, even in our single cell data, in the naive, the memory, the antibody secreting, is TNF signaling through NF-kappa B, in addition to the estrogen response pathways. I will point out that in our males, and I think this is interesting given some of the discussions that occurred earlier through the Stanford score, is that we do see some metabolic pathways being upregulated in males, so something interesting there. But type 1 and type 2 interferon pathways consistently come up, not just in our younger adults, but when we look in our older adults, we actually have no pathways that are enriched to a greater degree in older adult females than males, but some of these same pathways, including interferon gamma and interferon alpha, are still upregulated in older males. So much like was discussed earlier, when we look with these volcano plots, and what I've shown here are a few of the genes for which we do find they have estrogen response elements in their promoters, so they can be actually modulated by that estrogen receptor mediated path binding to these estrogen response elements. And what we find is greater enrichment of genes, particularly along this TNF signaling pathway, of genes that have these estrogen response elements in females, again in purple, as compared with males, and fewer of these in our older adults. So much like what was described with thinking about how to use organoids, we've been thinking about how to use our peripheral blood mononuclear cells to ask more mechanistic questions about estrogen signaling in B cells. And so if we take these cells and we do an ELISpot, where we are stimulating ex vivo with flu antigen, so that HA antigen, and we can do this for men and women. We can do this just in untreated cells where we're just taking and plating those cells. We can do this in cells that we actually stimulate with estradiol. Or we can do this in cells that we pre-treat with an estrogen receptor antagonist, tamoxifen, and then stimulate with estradiol and see what that does to the propensity of these cells to secrete antibody in response to flu antigen. And so that's what I'm showing you here in these ELISpot data. So if you just take cells from men and women, young adults, cells from females, they secrete more antibody. We get more spots, okay? So that's what I'm showing you here. And those spots are bigger, so on a per cell basis, they're secreting more antibody. But if you give those cells estradiol, we observe an increase. Not so much in the number of spots, but on a per cell basis, those antibody secreting cells start secreting more antibody. If you block the receptor with tamoxifen, you get a significant reduction. What I do find remarkable is the propensity for these cells to secrete more antibody, even among older adults, is there, even though that didn't translate into greater antibody responses to the flu vaccine. But we don't see the same effect of estradiol or blocking the receptor, so that does seem to be limited to our young adults. For the sake of time, I'm just going to point out that several genes along the TNF signaling pathway in young adult females, 7 days after vaccination, that have estrogen response elements are actually responsive to estradiol. We see an increase in the expression of these genes in B cells that have been stimulated ex vivo with estrogen, and a reduction if the estrogen receptor has been blocked. And if we take those B cells and we stimulate them ex vivo with flu antigen, okay, again, that hemagglutinin, what we find is that that estradiol increase that we see in the presence of HA, that increase in antibody secretion, both in the number of antibody secreting cells, but let's go over here to spot size, the size of those spots on a per cell basis. That increase I already showed you, estradiol really gets those B cells secreting antibody. I'm going to point out that in the absence of signaling through either NF-kappa B or TNF, estradiol cannot increase antibody secretion. So we know that that estrogen response signaling pathway is interacting with that TNF signaling pathway through NF-kappa B, that's required for estradiol to really have its effects. And the last point that I'm going to make with you, again, how is this biologically relevant? Well, I will tell you that when women have gone through menopause and are not on any type of hormone replacement therapy, they do in fact have lower antibody responses to that H3N2 component of the vaccine antigen as compared with young adult women who are premenopausal. We see that both prior to and after vaccination. And you know, I'm not going to belabor some of these points, but we can multiplex, we can look at a lot of different aspects. And for the immunologists in the room, we've become very interested not only in how estrogens, or maybe a loss of estrogens, can impact that antibody response, but how it can actually impact the binding of antibody to Fc receptors, which are located on innate immune cells, to turn on those innate immune cells. We heard about neutrophils. We care about neutrophils. We care about monocytes. We care about NK cells. And what I want to just bring your attention to is that when we look at the activation of these Fc receptors, premenopausally, you see a greater proportion of these Fc receptors being activated in blood samples that have come from premenopausal as compared with postmenopausal women. So we think not only is it about the FAB region of that antibody molecule, but that FC region could be equally important. And I'm just going to end with that. I think the public health significance of this is thinking about sex-based precision vaccinology, how we might use some of this information, some of these pathways to better understand how we might modify vaccines, including through use of adjuvants, and how that might be differed depending on whether we're male, we're female, as well as our age. And so with that, I'm going to end and thank you for your time.